All right, so the last learning objective for section 4.3 are prokaryotic structures. So we're going to look at this like we did with the eukaryotic cell. The plasma membrane is still going to be the defining characteristic between the external and the internal environment. These are the 5 things that are going to make up our external structures of our prokaryotic cell. We've already talked about a cell wall, but now we're going to start talking about a capsule, fimbriae, pili, and flagella. Internally, we have 2 new things. We still have chromosomes that will contain the DNA. We have another smaller unit that is separate called a plasmid. This is also a structure that will carry some of the DNA. This will have somewhere between 1,000 or 3,000 genes. This will only have 20 to 25. You have ribosomes, we've already talked about the cytosol, and then we have what are called inclusions or granules. So the bacteria, as they pick up extra nutrients, they're going to hold on to them because you never know when you're going to run into a good food supply that also has what you need. So when you find a good food supply, stock up. We're going to put them in what are called inclusions or granules. Remember, what's different about these is none of these are membrane bound. So this is just going to be a pile of salt, just like you would see in the domes when they have when they salt the roads. So we're going to talk about all of these structures. The ones that I've put stars by are the new structures unique to prokaryotic cells. You're going to need to also know the functions of all these things. I'm not going to talk about the functions of the things you know, the cell wall, the chromosome, the ribosomes, the cytosol, or the plasma membrane. You can go back to cell 1, the very first lesson we did, to find those structures. So let's begin. I'm going to do the plasma membrane in green. This is a structure you already know. Typically, when we think of bacteria, we think of these rod-like cells, but they come in all different sorts of forms. So this is my plasma membrane. We've already talked about the functions of the plasma membrane. The plasma membrane is the defining character between the inside and outside. It serves as the boundary. It regulates what comes in and out of the cell. It's there for protection, support, gives shape, lots of different things we've talked about. External to the plasma membrane is always the cell wall. So I'm doing this one in black. The cell wall will always be the first exterior structure, just like we saw in plants. But bacteria, they're going to add a third thing, and this is called a capsule. So a third structure called the capsule will always be exterior to the cell wall. This will also take on many different forms, but we have 2 major things that it does. It's going to help to protect the cell. This is something that makes sense. Capsules are typically thought of as hard, although even when they are not hard, they can also be very protective. These can help the cell to adhere to a surface. So if you've ever wondered why the bacteria can be 3/4 of the way up your shower wall, the capsule is one of those things that is helping it to stick to that surface. Because the capsule is generally the outermost surface, it also is going to serve as a sensory unit. This is the level that is in contact with the environment. So this is going to be the first thing that is going to sense the environment. And then the capsule can often be the defining thing for what gives something shape. So like I say, when you get to microbiology, you're going to learn there are lots of different shapes. It's not just this typical pill-shaped structure. So plasma membrane, cell wall, capsule, always in that order. Coming out of the plasma membrane for some bacteria can be a very long tail. This is called the flagella or the flagellum singular. I don't care which one you use. As you might guess, this is like a propeller. So this is going to have the function for movement. Now these are not the only hairs that come out. There are other structures that are projecting out. The short ones, so you notice flagella, you're only going to have one or a few. Typically they come out of one end. They can also come out of both ends. If you have the longest string that you see, that is always going to be the flagella. And like I say, there will only be a couple. But there are other hair-like ones that typically cover the cell. They also emerge out of the plasma membrane. But they're short and there's lots of them. These are what are called fimbriae. So all these short little green hair-like structures are fimbriae. They're another thing that will allow and help these cells to adhere to structures. So between the capsule and the fimbriae, they're both going to allow a bacteria to stick to a surface. And this is very important if you're a bacteria, especially if you're a pathogen, because step one is you got to get into the individual or your host. But then two, you have to figure out how to stick to them, because if you don't stick to them, your next breath out, you're just back out, and that does you nothing. So there's two ways that they adhere. This way they have more possibilities of sticking with their host or whatever substance they're on. All right, so we've got the cell wall, the capsule, we have the flagella and the fimbriae. A lot of teachers will mistake these hair-like structures and they will call them pili or a pilus. A pilus, though, is a tube-like structure that will come out. So I'm going to draw it like a tube. The pilus is generally a little bit thicker, typically not as long as the flagella, and typically coming out of the sides, whereas flagella are typically coming out of one end or the other. The pili is very important for what we would call bacterial sex. Now, bacteria don't have males and females. They don't have differentiating genders, but they can still exchange DNA. And the pili allow for bacteria to exchange DNA between different bacteria. We use gametes, sperm and egg. That's how we exchange DNA, and we recombine, we get new patterns. And this is a process that is called conjugation. So you've heard of prisoners having conjugal visits. This is when they're allowed to see their spouse and carry on marital relations, exchanging DNA. So the exchange of DNA is what will happen in pili. So these are my external structures. To give myself some room, I'm going to erase this so that we can talk about the internal structures. I'm going to leave the pili because I'm going to come back to it in a minute. The predominant structure inside a bacteria is going to be the bacterial chromosome. This is found in an area called the nucleoid region. The nucleoid is not a structure. The physical thing that is there is the chromosome. This area where we find the chromosome is the nucleoid. If the chromosome was over here, that would be the nucleoid region. So it doesn't matter where the chromosome is, but wherever it is what we call the nucleoid region. The bacterial chromosome has the same function as your DNA, so you can go back to that first cell one process and you can look it up. All of this is emerged in the cytosol. So remember, the cytosol is the fluid region. So if I have an arrow pointing to what looks like nothing, then that is the cytosol. We mentioned that all living things have ribosomes. The smallest units inside of a cell, whether it's prokaryotic or eukaryotic, are the ribosomes. And I point this out to say that they are the smallest because they can get confused with those inclusions or granules. Inclusions or granules are going to be solid because it's a big storage unit of materials. But look how much bigger this granule or inclusion is compared to a ribosome. So inclusions or granules, these are storage structures. So let's say you find a whole bunch of salt. So you're going to bring it in, you're going to glom it all together and let it just sit in the cytosol. So you find a whole bunch of fat. Great energy source. Bring it in, put it all together, let it sit in the cytosol. You can break it down later as you need it. So smallest units, ribosomes. Large solid units, inclusions. And then I mentioned that there are other small circular units of DNA that are called plasmids. So look, the bacterial chromosome is huge. This plasmid is really small. So the plasmid is just more DNA. These are bits of DNA, maybe 25 to 30 genes. So the bacterial chromosome, like I said, is thousands of genes. I mention this again because remember our pili, they exchange DNA. A bacteria is not going to exchange this whole lump of chromosome, but these plasmids can fit through the tube, and so that's how they exchange. So bacteria, they swap little bits of DNA, whereas we do half and half. These plasmids are maybe only 1 or 2% of the genome. So when you exchange DNA, we're talking about the DNA that is on the plasmids. So the DNA being exchanged by bacteria are these little snippets. Now, another thing I want to point out, when you're looking at and you're doing the homework, you're going to have to identify some structures. Inclusions and granules are circular, but they're solid all the way through. My plasmid is also circular, but you notice it's not solid all the way through. The DNA is just a piece of string and it's hollow in the middle. So when you're looking at your diagrams to tell an inclusion or granule from a plasmid, is it solid or does it have a hole in it? So these are the structures of our bacteria, or an archaean. A single cell prokaryotic. Much simpler than our eukaryotic. Matter of fact, I only gave you a total of 11 structures. I think there was at least 6 or 7 things just in the nucleus alone of a eukaryotic cell. So you can see how much the complexity has gone up. Now, I've also given you a few things that are not in your book, but I've given you a diagram from a microbiology book because you're going to see this again when you get to micro, and I want you to be prepared for a little bit more complexity that will come later.